Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor for today's event, Wyatt Technology Corporation, I'd like to welcome you to Molar Mass, Size, Charge, and Interactions, the light scattering toolkit for essential biophysical characterization. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. I would like to introduce our presenter for today. He is Dr. Daniel Sohm, Principal Scientist with Wyatt Technology. Welcome, Dr. Sohm.
Thank you very much, Elizabeth. Good morning to everyone. At least it's morning here in California. Whatever time zone you be in, it's good to have you on board. Today we'll be looking at an entire suite of tools that Wyatt Technology provides utilizing light scattering. Light scattering, it turns out, offers a variety of options for characterizing biomolecules. And we'll go through that suite of tools, that toolkit, and help you understand what can be done with it. And perhaps some of these can address the problems, the analytical challenges that you've been running into. So if you're involved in biomolecular research and development, you may be familiar with this R&D cycle, where along the way, especially if you are in the analytical business, you need to find out what your material does, what your biomolecules are doing. So you want to know, for instance, did I make what I intended? So does it have the appropriate properties, molecular weight, size, conformation? How is it aggregated? What is its conjugation state? After you've validated that this is the appropriate molecule, is it actually functional? So does it do what it's supposed to do? How do the interactions take place with targets and effectors? If it passes all those tests, you might want to check if it's actually well behaved. Does it self-associate? Does, when you go to a high concentration, for instance, with a monoclonal antibody, do you have viscous solutions that make it difficult to process or to inject subcutaneously? What are the interactions with excipients, which are designed to help keep it stable in solution? And finally, is it a robust product? So is it stable? Does it have a long shelf life? If you make multiple lots of it with different processes or different facilities, how do those lots compare? And once you've gone through the cycle, you may actually need to reiterate as you identify issues that come up, attempt to fix them through protein engineering or through changes in formulation and excipients or in a process. And some of the tools that will help you accomplish these are these acronyms, which I've mentioned over here, the SEC models and so forth. And as we go through the presentation today, we'll address these different acronyms so you know what they are. But just to get you started, each one of these is a technique that combines one form of light scattering with some type of sample preparation. So is there a separation going on upstream of the light scattering analysis, or is it measured as a function of concentration, or is it just measured over a series of different formulations and high throughput? And all of these are questions that we will address throughout the presentation. So first, I'd like to describe the light scattering techniques available to us in putting together these light scattering tools. The first one is multi-angle light scattering. Multi-angle light scattering is fairly straightforward. You have your sample in solution over here, and you hit it with a laser beam, and the laser will scatter off of the molecules, the proteins or the other nanoparticles in solution. And some of that scattered light, some of those scattered photons will reach the detector. In principle, our instruments measure the light scattering as a function of angle. That's not absolutely necessary for certain molecules, for smaller molecules, but it's definitely very important when you're looking at larger molecules because there is an angular dependence and there's information in that angular dependence. But the primary information that we're getting is actually the intensity that is being scattered by the molecules. And it turns out that there is a first principles relationship between the scattered intensity and the molecular weight and the concentration in grams per liter of the molecules in solution. So essentially, if we're measuring the scattered intensity and we measure or know the concentration, we can determine directly the molecular weight. This is a first principles determination, does not depend on any assumptions regarding conformation or other properties of the molecules. And so therefore, it's extremely useful and powerful tool. As I mentioned, the angular dependence also encodes information regarding size, so we can determine through that radius Rg, which is relevant for molecules with diameter of about 25 nanometers and larger. But for all molecules, we can determine molecular weight. Because it is a first principles determination, it also allows us to characterize interaction parameters. So for instance, stoichiometry of complexes, whether it's a hetero or homo complex, The binding affinity is Kd. In some cases, we can look at rates of association or dissociation, and also measures of nonspecific interactions between molecules in solution represented by second virial coefficient A2 or B22. So because this is a first principles analysis, it offers in-depth characterization, primarily looking at molecular weight. The second flavor of light scattering is dynamic light scattering. And dynamic light scattering, rather than looking at the average intensity that's scattered by the molecules, actually looks at the fluctuations of the light scattering intensity, which you might think of as noise. But that noise actually encodes some very useful information. And the reason that you see fluctuations is because of the Brownian motion of the molecules. So they're jittering around in solution under Brownian motion, and their rate of diffusion depends on their size. That relationship is encoded here in the Stokes-Einstein relationship, so diffusion coefficient, viscosity of the solution, temperature, and from this we can extract RH, the hydrodynamic radius, a measure of size. So how do we determine dt, the diffusion coefficient? That is actually encapsulated in the rate of fluctuation. So the smaller molecules will diffuse more rapidly. They'll have faster fluctuations. Larger molecules diffuse more slowly. They'll have slower fluctuations. And that entire spectrum of time constants of fluctuation is encoded in the fluctuations in the light scattering intensity. Through this analysis, we can extract the diffusion coefficient and therefore a hydrodynamic radius. So this is not a first principle measurement of molar mass as we get with multi-angle light scattering, but it is a first principles measurement of size. One of the really nice things about dynamic light scattering is that even without doing an upstream separation or fractionation of the molecules, you can get a coarse size distribution. In terms of interactions, dynamic light scattering is useful for extracting information about the nonspecific interactions in a formulation. And that's through the concentration coefficient of diffusion, which is known also as Kd. It's lowercase k versus the uppercase K on the binding affinity, and is in fact directly related to the osmotic virial coefficient. Another really nice aspect of dynamic light scattering is because it can be done in batch mode, and it's not quite as sensitive as multi-angle light scattering to the geometry of the cell and stray light and so forth, it's amenable to high throughput screening. And so we'll see in our light scattering tools how dynamic light scattering can be applied to high throughput screening of formulations and protein quality. There's the third technique that uses light scattering for analyzing macromolecules and nanoparticles, and that's phase analysis light scattering or electrophoretic light scattering. And what we're doing there is we're applying an electric field and observing the motion of the molecules under the applied electric field. So through essentially Doppler analysis of the light scattering, we're determining the speed of the molecules and divide that by the applied field, and we get a measure called the electrophoretic mobility. If we combine the mobility with the information on the size, the hydrodynamic radius, we can calculate what's known as the zeta potential or the net charge on the molecule. The instruments, one of the latest advances in instrumentation for PALs is MP-PALs, and that replaces the standard avalanche photodiode, which is generally used at a single forward angle, with 31 low noise and high dynamic range photodiodes collecting information at multiple angles. And that arrangement actually provides highly increased sensitivity, which means that you can reduce the measurement speed and you can reduce the applied voltages and therefore preserve your fragile biological samples. So how do we combine these light scattering techniques into what I call the tools, the light scattering tools? The first tool takes multi-angle light scattering and combines it with size exclusion chromatography. So remember, multi-angle light scattering is giving us a measure of molar mass and size. And by separating upstream based on size, we can actually determine accurately the distribution of molar masses within the sample through chromatography. The setup includes a multi-angle light scattering detector. Typically includes a concentration detector based on refractive index. Remember that in order to determine molar mass, we need the light scattering intensity and the concentration. So refractive index is a universal concentration detector. It's useful for proteins and for polymers and for all types of materials. We can also use the UV detector to determine the protein concentration if our LC has one. It's also possible to embed within the moles detector a DLS module so that we can determine simultaneously the molecular weight and the size of proteins which are smaller than the 10 nanometer limit for determining size by multi-angle light scattering, dynamic light scattering actually measures down to less than a nanometer. So the properties measured with SEC mols are distributions of absolute molecular weight, absolute because we're not depending on the elution time to determine molecular weight, we're actually measuring it directly, and size, often used in protein studies and biotechnology to look for small aggregates and oligomers, so the soluble aggregates. It measures molecular weight over a range of 200 Daltons up to, depending on the instrument, 10 or even 1,000 megadaltons. RH can be measured, again, depending on the instrument, between 1 and 300 nanometers, which is, in fact, well beyond the limit of separation of size exclusion chromatography. I'll describe a technique which uses this setup to analyze protein conjugates. So, for instance, PEGylated proteins or ADCs. By combining the information on molecular weight and the size from the online DLS, we can estimate the conformation of the molecule. Is it unfolded or is it compact? And the RG, which is the size that we determine directly from the angular dependence of mols, measures over 10 to 50 nanometers, or essentially the SEC limit, depending on the instrument. So why would you want to go to all the additional complexity of adding multi-angle light scattering to your SEC setup? Well, when you do analysis by SEC, you're making some assumptions about your sample and your references. You're assuming that the sample and the references have the same conformation. So, for instance, if your reference molecules are all globular proteins, and your sample is a globular protein, then probably it's the references represent well the elution time corresponding to the molecular weight. But if your samples are slightly different, then that's not a good representation. They assume the same density, essentially the same specific volume. They assume the same column interaction. So if your molecules are slightly hydrophobic or otherwise interact differently with the column packing material than the reference sample does, then again, the elution time is not a good representation. You're also assuming reproducible chromatography. So day to day, is your column doing the same thing? Is it aging over time? If things are happening which change the elution time as a function of molecular weight and size, then you'll need to recalibrate in standard chromatography. But the light scattering analysis doesn't really care about the actual elution time, and if it elutes a little bit later or a little bit earlier, it will still give the correct molecular weights. And that's illustrated here in this comparison of elution times and light scattering from BSA and hemoglobin. So BSA is one of our reference standards for elution, and you can see that we have the monomer and the dimer, and these two peaks elute at the times that you would expect based on the molecular weights. The light scattering, which is these dots, gives the molecular weight, and it shows, in fact, that this peak is highly monodisperse, so flat molecular weight across the peak. Same thing for the dimer, and it comes out at exactly twice the molecular weight of the monomer. Now look at hemoglobin. It elutes at a much later time, and you would expect, based on column calibration, that means that the hemoglobin is a much smaller molecule than BSA. In fact, that's not true. The molecular weights of hemoglobin and BSA are not too different. We see that in the light scattering. So here's the molecular weight from light scattering, and you can see that it is, in fact, pretty close to that of the BSA. You also get additional information from the mol's molecular weight, and that is that this funny-looking shoulder at the late elution times of the hemoglobin indicates a decrease in molecular weight, again, directly indicated by the light scattering. In fact, the reason for this decrease in molecular weight is the dynamic equilibrium between the constituents of the hemoglobin, which is, in fact, a homotetramer, and this indicates dissociation between the tetramer and dimer states, which occur as you go to as the hemoglobin undergoes dilution as it's going through the chromatography. So this indicates how important it is to have independent measurement of molecular weight, which does not rely on elution time. Here's another example of insulin in two different formulations. If we look at the red trace of the chromatogram, and then we see that it's primarily eluting in the monomer state, and light scattering gives us a measure of molecular weight, 5.8 kilodaltons, and also tells us that this little bump over here, around 14 milliliters, corresponds to a hexamer. In the other sample 2, which is a different formulation, we can see that primarily we're eluting in a much larger molecule, which light scattering tells us is a hexamer. And then there's a little bit of this later eluting peak. And if we look at the molecular weight across this peak, we see that it varies between monomer and dimer, indicating that this is, in fact, the monomer-dimer equilibrium. So very powerful information that we extract from the light scattering, not immediately apparent if you're just relying on elution time. MALS is also used for looking at much larger proteins. So for instance here, IgM and apoferritin with molecular weights in the range of the millions, including their aggregates. There are oligomers and aggregates, which you can see here going up into the tens of millions. An interesting thing to note about this is this peak of the IgM at around 17.5 minutes, and this peak of the apoferritin, which is about 19 minutes, they elute at very different times, but they have this almost the same molecular weight. So again, the MALS captures this information because it doesn't depend on elution time. Light scattering is particularly sensitive to aggregates. Remember that the intensity is proportional to molecular weight times concentration. So if the molecular weight increases greatly because you're looking at aggregates, then the concentration at which you can detect these molecules with light scattering decreases greatly. And so here is a peak in the light scattering, which does not show up in the concentration trace at all. And that's because it's high molecular weight aggregates. So again, the light scattering is particularly useful for identifying these aggregates. Another really powerful technique available with light scattering is the protein conjugate analysis. So if your species consists of two constituents, a protein and a modifier, for instance, polyethylene glycol or glycans, we can use a more advanced technique using three detectors to determine not just the overall molar mass of the molecule, but also the molar masses of the constituents in the molecule. So the experimental setup includes the MALS detector, the RI detector, which remember is a universal concentration detector, determines concentration of both constituents, and the UV detector, which in many cases will only indicate the concentration of the protein. For instance, PEG or glycans don't absorb in the UV. And so by combining these three, we can actually determine what is the concentration of the protein, what is the concentration of the modifier, get their ratio, and by adding the information from light scattering, determine the molecular weight of each of those in the bound complex. And so here's an example where we see across this peak, we've determined the molecular weight of the protein and of the modifier, and then the total complex, which is the sum of those. Here's the total mass. And in this example, we're looking at differences in host cell glycosylation, so expression in a mammalian and an insect cell. The protein molecular weight determined using the protein analysis, which is a feature of the Astra software, comes out identical for both of these expression cells. However, the carbohydrate molecular weight is different, so different degrees of glycosylation on the two types of host cells. And you also notice that the molecular weight varies across the peak, and that's an indication of the heterogeneity of the glycosylation in each of these systems. So there is some degree of different amounts of glycans binding to the proteins. Those are separated on the column, and you can see across the peak the actual distribution of those molecular weights. The protein conjugate analysis is also useful for analyzing antibody drug conjugates if you want to know the drug to antibody ratio, the DAR. Again, similar analysis. We use the three detector system. And here we've looked at two different ADCs, ADC1, ADC2, determine the total molecular weight of the complex, the molecular weight of the antibody alone, which is identical for both of them because they're based on the same antibody. And then finally, the molecular weight of the conjugated drug. This is actually the total molecular weight of the drug and linker, which is conjugated to the antibody. You'll notice this is a logarithmic scale, so these are very different. And then by analyzing down here in the table, the molecular weight of the antibody, the molecular weight of the drug linker, if we know the molecular weight of the individual drug and linker molecules, we can then determine the ratio, the drug to antibody ratio here. This analysis is quite universal, though I would say that the sensitivity is not perhaps as good as mass spec here. You need to have about 3 to 5% of conjugated drug linker. In other words, the mass of the total conjugated drug linker should be about 3 to 5% in order to get a good analysis. Here's another example of why it's really important to have both the multi-angle light scattering and the quasi-elastic light scattering, or dynamic light scattering, in line simultaneously. So in this chromatogram, we see 3 peaks, and nominally you might think that this is a monomer and some oligomers. But in fact, the moles analysis tells us that each of these peaks has the same molar mass. So what's going on? Well, there are some different possibilities as to why the same material with different molar mass might elute at different times. There might be some different conformations. So some of them might be unfolded or folded differently. Some of them might have some, hydrophobic moieties exposed, which would cause them to interact differently with the column. So which one is it? The answer comes from the inline dynamic light scattering. The dynamic light scattering is looking at the same scattering cell as the multi-angle light scattering, and so it's determining the hydrodynamic radius exactly at the same time in the elution as we're measuring molar mass. And what we find is that across this larger peak, we have a smaller hydrodynamic radius, and then this secondary peak has a larger hydrodynamic radius, indicating that this material is partially unfolded. And actually, the signal is not large enough to determine the hydrodynamic radius for this peak, but we can for this. And that tells us that unfolding is the culprit. As you may know, size exclusion separates based on volume, not based on mass. And so with a larger volume, but the same mass, will have an earlier elution time, which is what we see right here. Light scattering is also really useful for understanding protein purification. So here is a primary peak that was eluting from a purification column. There's a little bit of a shoulder down up here at about a little bit later after that peak, and then there's also some earlier peak which might correspond to some form of oligomer. So what are they? Is this tailing due to interaction with the column, or is it contamination, some other species which is present in the solution? Light scattering tells us, first of all, it tells us which oligomers we see here. So this peak, even though it's actually very well separated from the primary peak, turns out that it's a dimer. And then there's also a bit of trimer here and some tetramer all the way up here. The shoulder, which is at the trailing edge of the monomer, turns out that it doesn't have the same molar mass as the monomer. So it is in fact a different species. It's a lower molecular weight. We call it a fragment. We don't know exactly the identity. It might be a host cell protein in this case. It might be a fragment of the primary species. Hard to tell, but it is in fact an additional species and not just tailing of the monomer. Now, when you inject different concentrations into the SEC, you hope to identify changes in the molar mass due to concentration. So in the case I'm showing here, we're looking at a couple of oligomers, and regardless of the concentration that was injected, the molar mass came out the same. This means that this is a very stable oligomer, and even if you go to much lower concentrations, it's not dissociating. So it's a tightly bound oligomer or a native oligomer. That's not always the case. Sometimes when you inject different concentrations, there's dissociation. There's a dynamic equilibrium between some oligomer and the monomer. And so depending on the concentration, you'll get different molar masses. The light scattering tells you this very directly. So for instance, if we look at the red trace over here, we see that the molar mass falls off across the peak, and that's due to the decreasing concentration, shifting the equilibrium towards the monomer and the lower molecular weight. And again, as we inject different concentrations, so here's a lower concentration, it has a lower molecular weight at the peak than the higher concentration, again, due to the equilibrium being shifted towards the monomer. So this all comes out very directly from the measurements of molecular weight using multi-angle light scattering, something which would be impossible to determine using standard SEC. If you're familiar with ultra-high performance liquid chromatography, UHPLC, you know that has certain advantages: higher resolution, reduced mobile phase consumption, and faster throughput. And we have recently announced the Microdon, which is the first moles detector for UHPLC. And it takes all of the benefits that I just described for utilizing moles with SEC to the world of UHPLC. So in summary, our primary tool for characterizing proteins and other biomolecules, SEC moles determines absolute molar mass and size. overcomes the limitations of standard SEC like column interactions, conformation issues, bad chromatography, provides positive identification of aggregated unfolded species and purities, and determines the conjugation state. Second technique, which I'd like to go through relatively quickly, is FFF mols. So FFF mols is another technique that utilizes fractionation or separation upstream of the light scattering. And it is a technique, it's your go-to technique when standard SEC is not appropriate for your molecules. So if your molecules are sticking to the column, or you need to measure over a size range which is larger than a standard SEC column, or it's important to minimize the interactions between the molecules in the packing phase, field fill fractionation allows you to address these with a more sophisticated separation system. It does integrate with standard HPLC hardware, so it uses the same pump as the analytical HPLC, the same auto-injector, the same UV detector, but instead of using a column for separation, it uses this channel. And in the picture here, we see the Eclipse, which is the control system that controls the flows going through this channel. So the properties that are measured by FFF mols are similar to SEC mols, but work over a larger size range. In fact, it covers between 1 and 1,000 nanometers, a very large dynamic range for separation. Briefly, how it works, there's an open channel. The sample flows through the channel. And what we do is we constrict the output of the channel over here, which forces some of the solution to go through this semi-permeable membrane. And that forces the sample a little bit up against the membrane. Depending on the size, the sample will back diffuse into the channel. So the smaller things diffuse higher up in the channel, and the larger things stay closer to the membrane. The channel flow, which is the elution flow, has a parabolic flow profile. So those things which go higher see a faster flow rate, and they elute first. The larger things see a slower flow rate, and they elute later on. This crossflow can be tuned to change your separation power. So you can optimize for, say, zooming in, getting very high resolution in a particular size range, zoom out and look at a very large range of separation. And you can even change it on the fly so that, over the course of your elution, you can change the resolution and optimally identify and characterize different size ranges all at once. So briefly, we have a much larger dynamic range. The separation is as good as or better than size exclusion. Can be optimized for different ranges of separation. There's no stationary phase, so there's very little shear, and this works under most native conditions. Eliminates adverse interactions with stationary phase. And FFF is recognized by the FDA as an orthogonal method to SEC for characterizing aggregates. And so here's an example where we've zoomed in and gotten very high resolution for antibody oligomers. And you can see a nice separation of monomer, dimer, trimer, tetramer, and so forth. This is with a small injection, so it's 15 micrograms, and this gives us actually beautiful separations. We can also zoom out. And so in this case, we've changed the flow conditions so that we're separating over a very wide range. And so you can see that we're going up into the tens of millions, very large oligomers, essentially. This is an example of separating some silver nanolipid complexes, and it nicely illustrates the sensitivity of the mols measurement downstream of the FFF. So this is some nanolipids which are coated with microsilver, and depending on the conditions, there are different amounts of microsilver bound. These are the FFF separations. So in the red here we have the nanolipid by itself with no silver. That's the red trace over here. And then different amounts of bound silver. And we can easily identify, based on the preparation conditions, how much silver is bound to the nanolipid based on the radius. This is highly repeatable, so if you do multiple injections, same material, they overlay perfectly. And so these very small differences that we see here are actually quite robust. And so if you need to look at nanoparticles or combinations of nanoparticles and macromolecules, FFF is really the ideal characterization and separation method utilized together with the light scattering. Here's a separation of a VLP, which is partially formed from the capsomeres. And so there's complete VLPs and some incomplete fragments. You can separate the soluble and insoluble components of your solution as well. In this example, we've zoomed in, again, to look at some VLPs and two different species, very close in molecular weight and very close in size, but very well separated by the FFF and very well characterized by the light scattering. The same conjugate analysis can be applied to FFF and to VLPs. And so here's an example provided by Brent Larson at Berlix, formerly Berlix. And it was packed with some DNA. So here the VLP was actually being used as the delivery vehicle for the DNA payload. And again, we can determine the molecular weights of the constituents. The total VLP molecular weight was 15.5 megadaltons. The protein, not including the DNA, was 14.5, and therefore the DNA was about 1 megadalton, which corresponds to the 1.6 kilobytes very nicely. You also see the heterogeneity across the peak showing up very clearly. So FFF, as I mentioned, is pretty versatile, and it also comes with a variety of options for separation channels. And so using the hollow fiber channel, you can separate nanograms of material, and all the way up to the semi-perforative channel, which is good for separating milligrams of protein. There's a wide selection depending on the particular needs of your characterization or preparative work. Summarize, FFF mols is your primary tool for molar mass and size of proteins and other macromolecules, or combinations of macromolecules and nanoparticles that aren't separated well on SEC. You can look at submicron aggregates as well, characterize these complex fluids. It's also possible to couple FFF multi-CPMS if you need to address elemental analysis. Now on to the third tool. The third tool is CGMOLs, composition gradient multi-angle light scattering. And this tool addresses a different aspect of biomolecules, and that is their interactions. So instead of separating the molecules and characterizing the individual species, we're going to keep them together and understand how they interact if they form oligomers or heterocomplexes through the changes in molecular weight. And therefore, it's using the same detectors, the multi-angle light scattering detector, concentration detector, and instead of SEC, we're going to use a preparative system called the Calypso, which is the composition gradient system. Essentially what it does is prepare aliquots by mixing together different quantities of the proteins and diluent and injecting them into the light scattering detectors. This is a label-free and a mobilization-free characterization of interactions. It is very powerful because of its first principles, and it works from simple to highly complex interactions. Measures KD of a range of sub-nanomolars up to millimolars. Primarily, we use it for looking at equilibrium, but it's also possible in certain cases to look at kinetics, for instance, kinetics of dissociation or aggregation, and also to characterize formulations for non-specific interactions, as well as the specific interactions that lead to protein-protein binding. Properties measured are affinity stoichiometry. And by stoichiometry, we don't mean just the stoichiometric ratio, that is how many moles of one molecule bind to the other, but the absolute stoichiometry or the molecular stoichiometry, how many molecules of one protein bind to the other protein, or how many come together to form a homoligomer. Reaction rates in A2, B22, which are the Vero coefficients. So briefly, how do we measure self-association? We prepare and inject a series of concentrations, and presumably when you're at low concentration, you're primarily looking at monomers, and as you increase the concentration, you'll have more and more oligomers forming, and so the molecular weight increases. That's reflected in the light scattering. And so here are some simulations that explain how this works. If you increase the concentration and there's no self-association, then you just have a linear relationship between the light scattering signal and concentration. But if you have formation of dimers or trimers, then you'll have higher light scattering corresponding to the higher molar mass due to the formation of those complexes. And depending on the KD, you also get different shapes of these curves. So by analyzing the shapes and the heights of these curves of light scattering versus concentration, we can determine both the association scheme and the KD. If you're looking at high concentration formulations, things get a little strange, and so the light scattering intensity actually reaches a maximum, starts decreasing as you approach the concentration in the range of tens and hundreds of mg per mil. That actually gives you additional information. You can determine not only the association properties, but also the repulsive properties. For instance, if you have charge-charge repulsion, if you have heart-sphere repulsion, all of these are reflected in the shapes of these curves, and the data can be analyzed readily. For heteroassociation, we mix together the two proteins, protein X and protein Y, in a series of different stoichiometric ratios. And again, as complexes form, the molecular weight increases. That's reflected in the light scattering signal. If the binding affinity increases, we'll have more complexes and a higher molecular weight, so that's reflected in the change of the peak height with KD. And depending on the stoichiometry, both the position of the peak and its height will change. So the position will shift depending on the stoichiometric ratio, but the height also increases because the molar mass of 3 to 1 is higher than that of 2 to 1, higher than that of 1 to 1. And if we had, for instance, a 2 to 2 interaction, it will peak in the same position as 1 to 1, but it will have twice the light scattering signal because the molar mass is twice that of the 1 to 1. So by analyzing the height and shape of these curves, again, we can determine the association scheme as well as the KDs. The system for carrying out these measurements consists of 3 computer-controlled syringe pumps, which inject through some filters and some mixers, and there's some degassers along the way as well. Prepare the aliquots and inject into the light scattering. The flow is stopped. And so if there are slow reaction kinetics, we'll actually see that in the light scattering signal. Sometimes the reaction is very fast, and we just see a rapid equilibration with no time constant. And this technique was pioneered by Atri and Minton, described in 2005 in Analytical Biochemistry, and we have since commercialized CG mols. To look at a large variety of association schemes, so standard monomer dimer, but also monomer dimer hexamer, monomer hexamer oligomer, octamer, isodesmic self-association, which is progressive self-association. Basically, lots of different models can be set up and tested, as well as heteroassociation models, so the standard monovalent 1-to-1 or multivalent 1-to-1, assuming equivalent binding sites. If none of these models work for you, you can set up models and test for self and heteroassociation simultaneously. You can tease out allosteric inhibition or cooperativity, and also look at multivalent interactions, which are quite complex, but can be analyzed with this technique. Just looking briefly at analysis of a dimerizing system, which is known that, so this enzyme needs to be dimerized for activity. It's known that the activity decreases with ionic strength. And so the question was, does the dimerization change with ionic strength? We injected a series of concentrations and analyzed dimerization across those. And so here's what we found. We found that at larger salt concentrations, in fact, the dimerization does depend on the salt concentration. And so there's a monomer-dimer equilibrium, which gets weaker as you increase the salt, hence explaining the lack of activity as you increase the salt concentration. At lower salt concentrations, we also found the equivalent of a monomer-dimer equilibrium, but because we're measuring with light scattering the molar masses, we found that the effective monomer in this association was actually a dimer of the enzyme. And so what we were seeing was the self-association of dimers to form tetramers. And then right at this intermediate concentration, we can analyze the system and actually determine simultaneously the KDs of both of those processes going on, a very tight monomer-dimer association, followed by a weaker dimer-tetramer association. Insulin, in the absence of zinc, self-associates as well, but if you don't have zinc, it doesn't form hexamers. So here's a concentration series with insulin, plotting the apparent molecular weight determined from light scattering. And if it were forming a hexamer, it should look like this dashed line. But in fact, it is forming a different association scheme. It's a progressive association forming dimer, trimer, tetramer, and so forth, with a KD of about 52 micromolars. And again, this is determined directly by analyzing the light scattering and finding the association scheme that fits the data best. Here's an antibody-antigen binding. So there are 3 parts to this method. We're looking at self-association using a single concentration series of the antibody and of the antigen, here thrombin, and then looking at heteroassociation using this mixing gradient. And so here are the data, and I'll briefly explain the separate parts. What you can see here is that when we're mixing the two and injecting, the flow stops, and then there is a slow equilibration, and this is the kinetics that we see here in the signal, but we're only analyzing, for our purposes, the equilibrium. So we'll take the data after the reaction is completed. So the opening and closing sections of the gradient give us information about molecular weight and self-association of the constituents. Here we determine the molecular weight of the antibody and that there's no self-association, and of the antigen, again, no self-association. If we look at the intermediate part, this is what we expect to see. This dashed purple line is what you expect to see for no interaction. In fact, there is excess light scattering signal that tells us that there is interaction, formation of complexes. And we can analyze the equilibrium data from this series. If we naively try to fit the data to a one-to-one model, we'd find that there's a poor fit. So the squares are the measured data. The black dashed line is the best fit for a one-to-one interaction model. Clearly doesn't fit very well. Then when you try to fit to a 2-to-1 interaction model, assuming equivalent binding sites, we get a very nice fit over here. And we also determine at every composition how much we have an equilibrium of monomers, 2-to-1 complex and 1-to-1 complex. That's determined here. And we've determined a KD of 9 nanomolars, which is very nicely comparable to the value determined by ELISA, 15 nanomolars for this system. And I will skip over this one, but there's a lot more information. You're welcome to request the slides and to go through them. So CGMOLs provides a reliable assessment of biomolecular self-association in solution without labeling, gives you affinity and the absolute oligomeric state, the absolute molecular stoichiometry of complexes, rich information on the complex interactions. And while I haven't discussed this, it is one of the very few techniques capable of analyzing interactions at very high concentrations, up to a couple hundred mg per mil of protein. Lastly, I'd like to speak about batch DLS, so dynamic light scattering, using either the DynaPro high-throughput dynamic light scattering plate reader, which measures size distributions of proteins, aggregates, nanoparticles in microwell plates. These are standard microwell plates, the same ones they use in other types of, say, spectroscopic plate readers. It has temperature control from 4 to 85 C, and you can even use a built-in camera to look at each well to see if you have any precipitates or contaminants or bubbles in the well which are disrupting your measurements. Another option is a cuvette-based instrument, which obviously is not high throughput, but it does allow it to use very small sample volumes, larger range of temperatures. And this instrument includes a dedicated static light scattering detector, so that in addition to size, you can measure molecular weight absolutely as well. DLS, batch DLS, measures coarse size distributions. It's not as well resolved as if you do an upstream separation with SEC, but it does give you distributions of sizes and aggregates. Useful for looking at colloidal stability, and that's through the concentration dependence of the light scattering, unfolding or aggregation onset temperature, and you can even measure viscosity with dynamic light scattering. I'll skip the theory part, but basically looking at the results, if you have what we call a multimodal sample, a bimodal sample, 2 species in solution of different sizes, the DLS analysis will give you actually the radius of each of these sizes. And so here's an example looking at aggregates, different formulations before, with and without sucrose, pre-lyophilization, and then reconstituted after lyophilization, looking at the different amount of large aggregates that are formed. And you can even detect a small shift in the average radius of the smaller species, which indicates formation of small oligomers, say dimers or trimers. So in order to get actual resolution between these peaks, you need to have a difference of 3 to 5 times in radius, which is, of course, it's about a factor of 100 molar mass. So we're not looking at very small differences in oligomers, but you can see shifts in the width of this peak and then the average value of the peak of just a few percent, which actually does give an indication of smaller oligomers. If you do it on a screen on a microwell plate, it's easy to set up the software to color code a heat map. And so we've coded this one to show in red those wells which have solutions which are monodisperse or monomodal with low amounts of aggregation. The blue have some moderate amounts of aggregation, so you can see a broadening of the lower peak and some addition of higher aggregates. And then the black ones are very bad. This is micron size aggregates, so these are very heavily aggregated. So very easy to just load up this plate, especially if you have robots, run it through the plate reader, and come back after an hour or two and have all this information in front of you. You can also run temperature scans on the DynaPro plate reader. And so here's an example of looking some different formulations and seeing how the onset temperature for unfolding and aggregation changes as a function of formulation. There's small shifts here in the T onset, which is determined by extrapolating this down to the baseline. Small shifts in the average radius, which tell you a little bit about the initial aggregation state. And you can also see that if you hold it here at 7 DC, depending on the formulation, you'll have different amounts of increase in size. And this is actually ongoing aggregation as you sit at this higher temperature. So again, all this information comes out of the simple scan. You load up a plate with the different excipients, different formulations, let it run. If you have many samples, it might be overnight. If you have a few samples, might just be over the course of an hour or two. Come back and have all this information ready to go. And I will skip the viscosity part. But it is possible to do a coarse viscosity measurement with dynamic light scattering, particularly useful if you're looking at high concentration monoclonal antibody solutions. The final tool I'll talk about is the mobility detector, the Mobius mobility detector, which measures electrophoretic light scattering. Will help you determine molecular charge, which is, again, important for stability of your formulations. It is compatible with an autosampler, so it has a flow cell, and you can set it up with an autosampler to load and inject a variety of samples automatically, come back after it's all done, and your data are ready to go. It applies very little voltage to the protein solutions, which means that you're basically not frying your proteins in the course of the measurement. And because it measures dynamic light scattering simultaneously with the electrophoretic light scattering, you can monitor in real time if there are any changes in your sample while this is going on. Finally, if you have high salt buffers, that's historically been a major impediment to electrophoretic light scattering of proteins. The Atlas system pressurizes the flow cell, prevents the bubbles that are formed due to electrolysis, and allows you to perform these measurements of mobility in high salt buffers. So the properties that are measured are the mobility, the sizes, because there is a built-in dynamic light scattering as well. The combination of these two give you the charge or zeta potential of the molecule. You can use it to determine PI in native solution to assess colloidal stability, and you can even look at chemical degradation under your native conditions. If there are changes in charge due to chemical degradation, these can show up. So here's an application. We're looking at heparin and chondroitin sulfate, which is a contaminant of heparin. It's a similar molecule, which is supersulfated, and that those additional sulfur atoms add charge to the molecule. And so by analyzing heparin over a large range of molecular weights and the charge measured using the MP-Pals, we're able to determine a nice relationship between the weight average molar mass of the molecule and its effective charge. The supersulfated molecule fell off of that graph by about 30%, and so it was clearly picked out as having a different chemical composition through the change in charge. Another nice example of these measurement is looking at dendrimers, different generations of dendrimers, depending on their constituents. The acetic acid here adds charge, and so different generations will have different ratios of charge to generation, again, picked up through these measurements. And these were made in very high salt buffers, 500 millimolar of sodium sulfate. So MP-Pals determines the ADC charge states. The true PI determination of, for instance, your monoclonal antibodies in the formulation buffer, also useful for liposome formulations and other types of nanoparticles, particularly when they need to be measured in environments which, say, represent physiological saline. So coming back to the slide I opened with, we learned a little bit about how we can apply these light scattering tools to the analytical challenges that you encounter in the course of biomolecular R&D. Did you make what you intended? How do you characterize your molecules for molecular weight, size, conformation, aggregation, and conjugation? Something about measurement of interactions through binding to targets and effectors. The behavior, does it self-associate? Haven't mentioned, but excipient interactions is another area that can be addressed. And for instance, using the dynamic light scattering plate reader, you can test a large variety of excipients. And finally, is it a robust product? When you're assessing self-life, you can take your samples and figure out which type of aggregates formed. Did it fragment? Do you have fragments forming? And lot-to-lot comparability. And with that, I thank you for your attention. Hope you've learned something new today and it's piqued your interest. If you do have questions, I invite you to contact me at my email, dsom@wyatt.com, or to visit one of these web pages, wyatt.com/solutions, to learn about the different tools and what they address, or read some of the literature in our library, wyatt.com/library. So thank you very much. At this time, I'm open for questions.
Thank you very much, Dr. Sohm. We have so many questions, we can't possibly get to them all, but I've got a few here that we can see how many we can get through. The first question, what is the sensitivity of mols and DLS, and what concentrations are required?
Okay, so mols is about 100 times more sensitive than DLS. The specification for DLS it depends slightly on instrument, but not too much. We specified it in terms of the concentration of lysozyme. So approximately 0.1 grams per liter of lysozyme is a sensitivity for DLS. Because the sensitivity is directly inversely proportional to molecular weight, if we're looking at a monoclonal antibody, it's 0.01 grams per liter of IgG. Mols is approximately 100 times more sensitive. In fact, it's probably about 1 microgram per milliliter of lysozyme. It's actually a little more sensitive if you're very careful about cleaning up your solutions. So less than 1 microgram per milliliter of lysozyme and less than 0.1 microgram per milliliter of IgG.
Okay, perfect. Next question. How much modifier do you need to be able to measure the protein fraction and modifier fraction with protein conjugate analysis?
So it's about 3 to 5% additional molecular weight. So if your glycans represent at least 3 to 5% of the total molecular weight of the complex, that's where the analysis becomes robust. If it's down in the range of 1 or 2%, it's not going to be as accurate.
Okay. Can one observe large conformational change with only the mini-DON, not DLS?
Generally not for proteins. So the range of sizes that can be measured with the mini-DON is radius of 10 to 50 nanometers. Most proteins are smaller than that. They're much smaller than that. BSA is about 4 nanometers. IgG is about 5.5 nanometers. So unless it's a IgG perhaps with a complete unfolding, you might begin to see it, but I wouldn't count on it. If you're looking at much larger molecules, then yes, you could see those conformational changes.
Okay. Let's see what else we have here. What range of hydrodynamic radius can be measured by DLS?
So that depends if you're doing a batch measurement or an online measurement. And it also depends on the instrument, of course. So instruments are slightly different. The typical lower range for measurement is between 0.3 to 1 nanometers in radius. For the online DLS, it's about 0.5 to 1 nanometers. For the plate reader, it's about 0.5. For the nanostar, about 0.3. The upper range is quite different for flow DLS versus for batch DLS. So first, let me address batch DLS. Again, depends on the instrument, but it's up about 1 to 2 to even 2.5 microns. So that's the radius. For flow DLS, it depends on the angle at which the detector is placed, the scattering angle, and on the flow rate. For the mini-DON, it's about 40 nanometers. And for the Helios, the DON Helios, where you place the DLS at a very high scattering angle, it can go up to 200 to 300 nanometers, depending on flow rate.
All right. Can you perform protein conjugate analysis when the modifier is a small molecule?
It really just depends on the total added molecular weight. So if you're adding enough of that small molecule, then the answer is probably yes. Again, it just needs to reach 3 to 5% of the total molecular weight.
Okay, and it looks like we have time for just maybe one more question. What temperature range can be screened with the DLS plate reader?
4 to 85 degrees.
Oh, that was fast. We have time for a couple more then. What is the smallest molecular weight that can be measured by mols, and what is the largest?
So the smallest molecular weight that we've measured with mols was a 200 Dalton monomer of a polymer, eventually a synthetic polymer. So 200 Daltons, the concentration needed to be relatively high, not too high. It was done on SEC. The highest molecular weight is up in the hundreds of megadaltons, even as much as a gigadalton. Okay,
last question. Are there any solvent or excipient limitations for DLS?
If so, Yeah, the answer is it depends. Excipients that have large molecular sizes, which are comparable to the size of the molecule. For instance, if you have micelles of detergent, which are comparable to the size of the molecule that you're measuring, those will definitely interfere with the measurement. Because they'll show up in the same size range, and it'll be difficult to distinguish which is which. If you're talking about small excipients, salts, no problem. If you're talking about something which is very large and kind of out of the, if you have very large excipients, for instance, alum adjuvants, those will scatter so much light that they will actually blind the DLS to the smaller molecules. That's when having a separation like FFF upstream really helps with the analysis.
Perfect. And we've just come on to 12 noon here in the East Coast. So I want to thank you so much, Dr. Sohm, for your presentation. I'd like to thank Wyatt for sponsoring today's presentation. I'd like to thank all of you who came today. We really appreciate you spending an hour of your time with us. And I wish you a wonderful day wherever you are. Thank you, and bye-bye.